Today we're going to begin by talking about the synapse. So the synapse is the area where we've got two neurons coming into contact with each other and communicating. So if we look at a synapse, we have a presynaptic neuron and a postsynaptic neuron. And when you're deciding what a presynaptic neuron and a postsynaptic neuron, which neuron is which, you're only ever talking about this synapse. So in this synapse, this would be the presynaptic neuron, this would be the postsynaptic neuron. Now this postsynaptic neuron would be the presynaptic neuron for the next neuron. So they're not always in a fixed position. You just determine which one is which based on the synapse that you're looking at. So our presynaptic neuron is going to be conducting an impulse toward our postsynaptic neuron. This impulse is the action potential. So then our postsynaptic neuron is receiving the signal and responding. So if we look at this electron microscopy, we can see that this is a presynaptic neuron that is synapsing onto the cell body of a postsynaptic neuron. And what you can see is this postsynaptic neuron has several presynaptic neurons synapsing onto it. So in real life, this postsynaptic neuron could have another synapse here, another synapse here, but we're only ever looking at one, just to simplify it. So we've got two types of synapses in the body. Chemical synapses, which are the most abundant, and this is going to be where the communication from our presynaptic to postsynaptic neuron is happening because of a chemical called a neurotransmitter. So our chemical synapses are the most common, and we'll say the signal is transmitted by a neurotransmitter. So an action potential is going to be fired in our presynaptic neuron. It's going to release a neurotransmitter to the synaptic cleft. It's going to cross the synaptic cleft and bind to its receptor, which is a chemical gated receptor on the postsynaptic neuron. This is the most common type of synapse we have. So maybe you can get it to work from the lecture. None of the animations are working from the computer here. I don't know why. But this kind of just shows you that if we look at our presynaptic neuron, it's got all of these synaptic vesicles with stored neurotransmitter. When our action potential comes down, these vesicles will fuse, the neurotransmitter will cross the cleft, bind its receptor, and open that channel. So as far as our chemical synapses go, again, they're the most common type. If we think about the way that the steps that are taking as information is crossing the chemical synapse, we can break it down into 6 steps. So the first thing that's going to happen is we're going to have this action potential that's spreading like a wave down the axon, and it's going to arrive at our synaptic knob. So the first step is the action potential is transmitted down the axon to the synaptic knob. If we look at the synaptic knob, it's got voltage-gated channels that are voltage-gated calcium channels. And when this action potential gets down there, it's going to open up those channels. So this right here would be a voltage-gated calcium channel. And calcium is always higher outside the cell than in. So when the action potential gets down here and opens this channel, calcium is going to come into the cell. Why is this important? Well, if we think about it, we'll kind of blow out the synaptic knob here. Our neurotransmitters are stored in vesicles. Vesicles are made of the same stuff as plasma membrane. So they can bind with the plasma membrane, fuse with it, and dump neurotransmitter. But we only want neurotransmitter being released when we've got an action potential coming down and the neurons being stimulated. So when you're at rest, there are all these cytoskeletal proteins that are holding these vesicles in place so they can't fuse with the plasma membrane. So that's how it's possible to hold these synaptic vesicles with neurotransmitter, is because these proteins are hanging onto them. So what happens when our action potential moves down to the axon terminal and we open these voltage-gated calcium channels, calcium is going to come into the cell and it binds with these proteins, shifting them away from the vesicles so that the proteins can let go and the vesicles can fuse with the plasma membrane and dump a neurotransmitter by exocytosis. So now it can enter the synaptic cleft, cross, and bind its receptors. So for the second step, we could say that our voltage-gated channels open and calcium is going to flood the axon terminal. So this figure in your book kind of shows that. We have this action potential that's coming down our axon. It's going to trigger the opening of these voltage-gated calcium channels. So they open, calcium enters the cell. Our next step is that calcium is going to bind with those proteins and allow the synaptic vesicles to fuse with the plasma membrane. So we'll say calcium entry allows vesicles to fuse with the plasma membrane and release their neurotransmitter by exocytosis. So the neurotransmitter enters the synaptic cleft. Then our next step is that it's going to cross the synaptic cleft and bind with its receptor. This is very highly specific. So whatever neurotransmitter this is can only bind to a receptor that's specific for it. So if it's acetylcholine, it's got to be an acetylcholine receptor. If it's dopamine, it's got to be a dopamine receptor. You can dump dopamine all day long on an acetylcholine receptor and nothing will happen. Receptor specificity is really, really important. So this binding is going to open an ion channel of some sort, and there will be some type of graded potential produced. So for our fourth step, we can say that our neurotransmitter crosses the synaptic cleft and binds its receptor. And this binding is very specific. And then the next step is the binding is going to open an ion channel and will generate some sort of graded potential. We briefly talked about graded potentials last week. We just said they decay over time, they travel short distances. We're going to talk in more detail about them today. So then the last thing that happens at the synapse, in a chemical synapse, is now, after we've had this binding and we have the graded potential develop in our postsynaptic neuron, as long as this neuron is no longer being stimulated, we need to remove the effects of it. So we terminate the effects of the neurotransmitter, and that can happen three ways. Either the sending cell can pick back up the neurotransmitter, or a nearby astrocyte can, or we can have enzymes break it down, or it will just diffuse away. So for our sixth step, we'll say we terminate the effects of our neurotransmitter. And this is going to happen one of three ways. What we call reuptake. And this will happen by the sending neuron or by an astrocyte. So we'll say reuptake by the presynaptic neuron or an astrocyte. Enzymatic degradation, like we saw in the case of acetylcholinesterase, when it breaks down the acetylcholine in the neuromuscular junction, would be the second way. Enzymatic breakdown, or just diffusion over time. All right, so that's how we transmit a signal across our chemical synapse. But if you think about it, then this process kind of takes a little bit. It's not really, I mean, it's fast, as fast as you can think, but it's not that fast. It's because we've got to get our action potential down here, we've got to have the fusion of our vesicles, our neurotransmitter has to cross the synaptic cleft, find its receptor, bind it. So it's not as fast as an electrical synapse. So they are most common by far, but they have what's called synaptic delay that happens because all of these events take a while. So our other type of synapse is an electrical synapse. In an electrical synapse, our pre- and postsynaptic neurons are joined by gap junctions, so conductivity is pretty much instantaneous. So these are by far less common. And we can say our presynaptic and postsynaptic neurons are joined by gap junctions. This makes them very fast. So if we were to look, our presynaptic neuron would synapse onto our postsynaptic neuron, and they'd be joined by a gap junction. So it's almost instantaneous communication from one to the next. You don't find these in very many places in the body because we want to be able to have a lot of different input coming into a postsynaptic neuron, and we want to be able to have some kind of specificity for whether or not we fire an action potential. So where you find electrical synapses are in places that we have to do super fast movement, like for moving your eyes. So eye movement happens very quickly, especially in response to reflexes. If something goes flying past your face, you're going to respond and follow it. So you find them in your eyes, and you also find them in this place called your hippocampus, which is where memory formation begins. So we also kind of need fast signaling there. But everywhere else, we kind of want to be able to sum up what's going on. So a good example, somebody asked, well, if chemical synapses are slower, then why do we use them at all? Well, let's say you stub your toe, and your toe really hurts, and you're thinking about how your toe really hurts, but then a guy comes in the room with a gun. That's more important to think about the guy and the gun and how I'm going to get away from him than it is to think about my toe hurting right now. So you can sum up the different input to that neuron, and you would be able to inhibit the pain signal and then start stimulating signals to get us fighting and fleeing. So our chemical synapses allow the brain to respond to multiple different types of stimuli at the same time. Does that kind of make sense? So it makes a lot more sense to have these chemical synapses because then they can sum up information from a variety of presynaptic neurons to determine what this postsynaptic neuron is going to do. Okay, so the next thing we're going to talk about is our graded potential specifically, because this is going to be how our postsynaptic neurons are determining whether or not to fire an action potential, is by summing up our graded potentials. So in order to understand all of this, I think it helps to look at a neuron and see what type of channels and pumps we have where, because it helps us understand why a neuron is behaving the way it does. And it's got lots of dendrites. So when we're talking about our graded potentials, then we're going to consider this our postsynaptic neuron. And the dendrites are the receptive segment. So and the cell body, I should say. The dendrites and the soma make up the receptive segment. So they can respond to incoming neurotransmitters. The axon is the conductive segment. It's going to conduct an action potential away from the cell body toward the axon terminal. So our axon terminal, or our synaptic knob, is the area where we're going to now transmit this signal from this neuron to the next. So this is our transmissive segment. We just talked about our transmissive segment, and we said that the channels that we have here are voltage-gated calcium channels. We talked about our axon last week, and we said that the important channels that the axon has are our voltage-gated sodium and voltage-gated potassium channels. So pictures generally only ever show voltage-gated channels on like the top or the bottom, but this is a three-dimensional structure, so they're found all around. And they're found in high concentration at the axon hillock. And then they're found between our myelin sheaths and myelinated neurons, or at little intervals down the rest of the length of unmyelinated neurons. So our voltage-gated sodium channels. So these are going to be responsible for the depolarizing swing of our action potential. The other channels that we have in very close proximity to our voltage-gated sodium channels are our voltage-gated potassium channels. In our receptive segment, we have neurotransmitter-gated channels. So if you think about it, if this is the postsynaptic neuron, it's responding to signals that are coming from a presynaptic neuron. The presynaptic neuron releases neurotransmitters. So all of the receptors that we find here are neurotransmitter-gated or chemically gated receptors, but the chemical is a neurotransmitter. So these are chemically gated receptors or channels, but it's a receptor, and then when it opens, it's a channel. So, and the chemical is a neurotransmitter. So we've got three types that we'll find in the receptive segment. Each of these different channels has to have a specific key to open the gate. And it's a channel, so once the gate opens, an ion is going to move down its concentration gradient. So, again, where is sodium higher? Outside. Where is potassium higher? Inside. Where is calcium higher? Outside. We only are looking at calcium here, but there's higher calcium out here too. It's just higher outside. This is interstitial fluid, so the whole thing is surrounded by this. Our resting membrane potential, again, is set by those sodium-potassium lead channels and the sodium-potassium pump. They are found down the entire length of the neuron, the whole entire neuron. So I can measure my resting membrane potential here, it would be negative 70. I can measure it here, it would be negative 70. I can measure it here, it would be negative 70. I can measure it here, it would be negative 70. Because those lead channels and the pump are setting that. So the specificity and the way that a neuron behaves is set by these other types of channels. And each part of the neuron can only respond to one thing. So let's look at that. Our receptive segment can only respond to neurotransmitters. And when a neurotransmitter binds its receptor, it's going to open an ion channel. We've got three types of ion channels. The first is going to give us what's called an excitatory postsynaptic potential. So our excitatory postsynaptic potentials are little depolarizing events that move us closer to threshold. So an EPSP is an excitatory postsynaptic potential, and these are our graded potentials that move us toward threshold. Threshold for what? The threshold for these voltage-gated sodium channels to open. As soon as they open, sodium is going to rush into the cell and begin our action potential that is self-propagating. So our excitatory postsynaptic potentials are these little graded potentials that move us closer to threshold. What's going to happen, or what's going to cause an excitatory postsynaptic potential, is that a neurotransmitter is going to bind a cation ion channel. We've already met a cation ion channel when we talked about muscle firing. Our acetylcholine binds a cation ion channel at the motor end plate, and we've got this movement of sodium into the cell and potassium out of the cell. It's a channel, so they're both moving down their concentration gradients. But more sodium is moving in than sodium moving out. So we could put some little blue channels on here, and these would be our cation ion channels, and these are going to let sodium in at a faster rate and more than potassium out. So if we look at the specific movement of our ions, then potassium is moving out, sodium is moving in, but this is happening at a much faster rate. So we've got sodium coming in, so since it's positive charge coming in and it's coming in faster than potassium out, we have the potential maybe to reach threshold. So that's our first type of chemically gated channel. Our next type of chemically gated channel is a chloride ion channel. And both of our other types of chemically gated channels are going to cause the next type of graded potential. So before we go to them, let's just look. If we have an excitatory postsynaptic potential, it's a small depolarizing event that happens in our postsynaptic neuron, and it happens because when the neurotransmitter binds, it's going to open this cation channel. We'll have a net influx of sodium, which will cause this little graded potential. If we don't reach threshold, we don't shoot an action potential. It's a graded potential. So the further we get away from the area of stimulus, the more it decays. We have what are called inhibitory postsynaptic potentials. So let's say our presynaptic neuron dumps acetylcholine. Acetylcholine most of the time is excitatory. It's going to bind its receptor and open this channel. Now sodium's going to come in, which is the most important part. because that's going to bring us toward positive. It's a cation channel, so both are moving. Potassium is moving out, but it's happening slower, and it's not enough to make it more negative. So that's going to cause an excitatory postsynaptic potential because positive charge is coming in, and that's going to move us closer to the threshold for these guys. Okay. So then the other types of postsynaptic potentials we have are inhibitory postsynaptic potentials, or IPSPs. And these are going to be, they're also graded potentials, but they're moving us away from threshold. So we're going to become more negative. So in this case, we've got two types of channels that can be opened for an inhibitory postsynaptic potential. So first, I'll draw this little one. This is a chloride ion channel. So when this opens, chloride ion will go down its concentration gradient, and it's higher outside the cell. When this happens, chloride ion, which is negative, rushes in. So now if we're bringing a negative charge in, that's going to bring us further away from threshold. It will be inhibitory. It's harder to get an action potential if we're bringing in a negative charge. So that's our first type of channel that will have an inhibitory effect, and that's because now we're bringing in negative charge. If we've got to get to threshold, which is negative 55 from negative 70, then this negative ion coming in is going to pull us further away from that. The second type of channel, we've got a second type of inhibitory postsynaptic potential. The second type of channel that can cause this is just a potassium ion channel. And all that's moving is potassium. So when potassium moves, it goes from high to low, from outside the cell, or from inside the cell to out. So if positive charge is leaving, that will also move us away from threshold. So the second type of inhibitory postsynaptic potential comes when a neurotransmitter binds to a potassium channel. And what will happen is potassium will exit. So this is also inhibitory. So this would be positive charge leaving the cell that also brings you away from threshold. So if we look at what an inhibitory postsynaptic potential looks like, they are going to drive the neuron away from reaching this action potential threshold. And what's happening then is our neurotransmitter is binding either a chemically gated chloride ion channel, which is going to bring negative charge into the cell, or a chemically gated potassium channel that's going to bring positive charge out of the cell. Either way, we are coming from our negative 70 and becoming more negative. So it's going to get us further away from threshold. So it's inhibitory. It's moving us away from the potential of firing an action potential. So we can have both types of postsynaptic potentials happening to a neuron. So it's got to, our neurons have to sum that information up to decide whether or not to fire an action potential. So summation is going to be how we sum up our inhibitory and postsynaptic potential and excitatory postsynaptic potentials to decide whether or not to shoot an action potential. But do you see how by having these different types of channels in different parts of the neuron, each neuron can only do really one thing? Our soma and our dendrites can get these little graded potentials, and if they sum up to threshold, all we have in our axon are voltage-gated sodium and voltage-gated potassium channels. So if we sum to threshold, these will open. Sodium will come in, it will depolarize the next one, sodium will come in, these are going to enter their inactivation state, the potassium channels are going to open, potassium is going to go out, so that's our depolarization followed by repolarization. That is the action potential. So as sodium comes in this one, it's going to diffuse down and stimulate this one to open, this one's going to enter its inactivation state. Sodium comes in, diffuses down, stimulates this one to open, this one enters its inactivation state. So there's no choice but for the action potential to go one way, away from the soma, toward the axon terminal. And then when we get to the axon terminal, the only channels that we have are voltage-gated calcium channels. So when the action potential gets down there, calcium enters, now we release our neurotransmitter and transmit our signal. If we had chemically gated channels here, then we wouldn't be able to have an action potential. If we had our sodium and potassium channels here, then we wouldn't be able to release our neurotransmitter. So the specific types of channels that you find in each part of the neuron are telling you what that part of the neuron does. And it's literally one of those times when the structure leads to the function. So this is when anatomy, one of the places where you can see anatomy fits physiology perfectly. Okay, So if we can have both excitatory postsynaptic potentials and inhibitory postsynaptic potentials, then the soma kind of has to add them all up. They've all got to reach threshold in order for the action potential to fire. So we've got two types of summation. The first type is called temporal, and that's when we're summing up our graded potentials in time. So first, let's say that summation is adding up our graded potentials to decide whether or not to fire an action potential. If we have all of these potential, we've got, you can have hundreds of presynaptic neurons synapsing onto one postsynaptic neuron. So you could have all sorts of information coming in. So it's the job of the soma to get all of those ions to the axon hillock and decide whether or not to fire that action potential. So in temporal summation, what's happening is we're receiving stimuli close in time. So temporal is summing up in time. So if I have, let's say this is an excitatory synapse right here, and acetylcholine is being released and it's binding to our cation channel. If this neuron fires and fires and fires and fires, then what's going to happen is sodium is going to come in. Now if it just fired once and that was it, sodium would come in, but then it'd be pumped back out. Our leak channels and resting membrane, or our pumps would take back over and reestablish. So in order for us to reach that threshold, we've got to sum up more than one excitatory postsynaptic potential in some way. So if this one's being stimulated a lot, and it's like releasing acetylcholine, releasing acetylcholine, releasing acetylcholine, now sodium can come in and it can diffuse. And then we get hit again. So more sodium comes in and diffuses. And then we get hit again. So more sodium comes in and diffuses. And you don't have enough time to reestablish resting membrane potential because our excitatory postsynaptic potentials are happening so quickly that we're basically hitting this channel over and over and over again. Acetylcholine is continually being released from the presynaptic neuron, which is keeping these channels open, and sodium will have time to diffuse in and allow these voltage-gated channels to reach threshold. That's how temporal summation works. Spatial summation is when we have like several potentials being added in different places at the same time, basically. So it'd be like I've got this one acetylcholine channel being hit there, but I've got another presynaptic neuron that's stimulating this channel here, and I've got another presynaptic neuron that's stimulating this channel here. So this one's letting in sodium, and this one's letting in sodium, and this one's letting in sodium. So that's all summing up in space. As long as they're all open, we'll have enough sodium coming in through our three different channels to reach threshold at our axon hillock. So spatial summation is summing up because the neuron is being stimulated at several different parts at once. So basically it's summing up in space. So this is showing us what happens with no summation. So we've got an excitatory synapse right here, but it's stimulating our postsynaptic neuron, but not fast enough to sum up. It's not getting hit quickly enough. So it's releasing a neurotransmitter that's allowing sodium to come in, but we're not reaching threshold. And then our postsynaptic neuron is going to come back to rest because of our sodium-potassium pump and our leak channels. And then it gets hit again, sure, but it wasn't close enough in time to sum up. In temporal summation, you have rapid excitatory postsynaptic potentials being delivered. So now we can see it's the same presynaptic neuron, but now it's stimulating it really quick in time. It's like 1, 2, instead of being like 1, 2. It's happening really fast. And so this first postsynaptic potential is going to bring us closer to threshold. It wasn't enough on its own. But that second excitatory postsynaptic potential, it's also just little, but it's summing up on top of the sodium that's still in there. And as soon as we reach threshold, our action potential is going to fire. So this is what's different about our graded potentials and our action potentials. Graded potentials are small and they decay over time. They can add up. As soon as we open that voltage-gated sodium channel, we will depolarize all the way to positive 30 millivolts. It has no choice but to behave that way. So that's temporal summation. In spatial summation, this would be when we get stimuli, both excitatory at different places, but it's happening at the same time, so they're still able to add together. So this E1 is our first excitatory synapse, E2 is our second excitatory synapse. As long as we're exciting the neuron at the same time from both of them, then this postsynaptic potential is going to be able to add with our next postsynaptic potential, and again, we'll fire our action potential. Now it's possible to also have inhibitory things going on, so that's the job of the cell body, is to kind of be adding all of these up. So if we have an inhibitory coming from this inhibitory neurotransmitter, and then this is also showing spatial summation, and one excitatory, if our inhibitory pulls us further away, then even though we have excitatory, we're still not going to reach threshold. So summation can work both ways. We could add up, and it could be temporal too. You could inhibit, then we go negative, 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 and get further away from action potential. Does that kind of make sense? So summation works both ways, and you can have hundreds of presynaptic neurons. So it's all about whether or not we're reaching that negative 55 threshold to open these voltage-gated channels. And if we do, then the action potential is self-propagating and it will conduct. You cannot stop it. It's an all or nothing phenomenon. As long as we open these channels, we'll depolarize to positive 30. Then we'll open these ones, we'll repolarize and actually hyperpolarize so that these can stay in their inactivation state while these depolarize. And then these will depolarize, and then this will hyperpolarize, so those go in their inactivation state. And it keeps it moving in one direction away from the soma, so that we can get down here to our voltage-gated calcium channels, which will allow calcium to move those proteins so we can dump neurotransmitter. And now this neurotransmitter will be excitatory or inhibitory to the next neuron. Okay, so here's this nice table that compares our graded potentials to our action potentials. Our graded potentials, they're located in different places. Our graded potentials are happening in the dendrites or the cell body. So they are only happening here in the receptive segment. Our action potentials are only happening in the axon. Other things about them, the amplitude as far as size goes, our graded potentials can be different in size depending on what's going on, depending on what kind of channel we're opening, depending on how they sum. Our action potential is always the same magnitude. It always goes from positive 30 down to negative 80 and then back to rest. Positive 30, down to negative 80, back to rest. So our graded potential can decay with distance. Our action potential does not. It is always the same size. It's an all or none phenomenon, and it doesn't decay over time. It always has the same magnitude. What's the stimulus for opening our ion channels that create a graded potential? Neurotransmitters binding to their receptors. So our chemically gated receptors are going to stimulate our graded potentials. Our action potentials are stimulated by opening our voltage-gated channels. Don't worry about this positive feedback thing, it's kind of weird. Repolarization in our graded potentials just happens because of our sodium and potassium leak channels and our sodium-potassium pump. So as long as there's not stimulus happening, the graded potential will decay. An action potential repolarizes because of our potassium channels. So repolarization in the action potential is due to our voltage-gated potassium channels. As far as summation goes, we have summation that occurs in our graded potentials. It can either be temporal, where we're happening really closely in time, increased frequency of stimulation, or spatial, where we're getting stimulation from multiple sources. And in action potential, there's not like summation from anywhere else. As soon as we hit threshold, we will fire our action potential. Again, it's all or none. And then as far as graded potentials go, we have two types: excitatory postsynaptic potentials that move us closer to threshold, and inhibitory postsynaptic potentials that move us away. Our graded potentials are short distances, they decay over time, they don't last very long. Our action potentials can go long distances, and they are self-propagating, and they are what's considered this nerve impulse that's being conducted. Our graded potentials are caused by three different types of channels: either an excitatory postsynaptic potential, which is being caused by this opening this cation channel, an inhibitory postsynaptic potential, which is caused by opening a potassium channel or a chloride channel. In our action potential, this is dependent on our voltage-gated channels. Our voltage-gated sodium channel, which is going to open for depolarization, and then our voltage-gated potassium channel, which is going to open for repolarization.